Okay, so let's start on the slide where we were at the end of lecture 3. So temperature sensitive mutation belongs to a class of mutations which are called conditional mutation. Okay, so as the English suggests, conditional mutation meaning in certain conditions they will behave as wild type, in certain other conditions they will behave as mutants. In this particular case, the condition is temperature, that at a lower temperature the cells will behave as wild type, and an elevated temperature the cells will behave as mutant. And the reason for that will be that certain proteins will not have a stable enough structure at an elevated temperature, therefore they will not be able to function properly. And that can be due to a variety of reasons. At an elevated temperature it unfolds and gets degraded. At an elevated temperature it changes its structure slightly, but badly enough that it cannot interact with the its interacting partners in the cell. There are so many reasons why it will not be effective at a different condition, which is the elevated temperature. But there can be other cases, other kinds of conditions can be used, okay, like in the presence of a small molecule drug or in the absence of a small molecule drug. So the cell will behave in a particular manner in the presence of a small molecule drug and will behave normally in the absence of that drug. Or in the presence of glucose or absences. Glucose is not a good example. In the presence of galactose or in the absence of galactose, that is another conditional mutation. So there can be so many different kinds of conditions. That's number one. Number two is that now whatever I told you in the last lecture in terms of temperature sensitive mutation is that you are just looking at generically, okay? The cells are not growing. There can be a variety of reasons why the colonies are not growing, okay? It can be that there is an acute defect in protein synthesis at elevated temperature. Possible, right? Some key members of protein synthesis pathway, if they have undergone temperature sensitive mutation, at an elevated temperature there will be no protein synthesis or very minimal protein synthesis, so cell will not be able to grow. It can be because the intracellular trafficking is blocked. Okay. So because you are only looking at the end phenotype. What is the end phenotype? That the cell failed to grow. Okay. There are, as you can imagine, hundreds of reasons why a cell may fail to grow. But you can also have a directed search. Okay. For example, imagine you are only looking for those genes which are important for our favorite example, galactose metabolism. You are looking for temperature sensitive mutation of galactose metabolism. So what will happen? These plates, which is marked here 23 degree, 36 degree, the same thing, but there will be, instead of 2, there will be 4 plates. That one pair will be 23-36 in glucose, one pair 23-36 in galactose. Those that failed to grow at an elevated temperature, both in the presence of glucose or in the presence of galactose, they are not specifically defective for galactose metabolism because they could not grow in the presence of glucose as well at an elevated temperature. They are temperature sensitive, all right, but they are not temperature sensitive for galactose metabolism. But a subset of them, no, no, like there will be a different set which will not be able to grow at an elevated temperature only when the plate has galactose as the sole carbon source. In the glucose pair at 23 and 36, they will grow well in both the cases. So they do not have a generic defect. Like generic defect meaning they can grow in presence of glucose. Okay? Even at lower temperature, at higher temperature. But galactose metabolism requires a specific set of enzymes that are not produced when you have glucose as a carbon source. Those enzymes are produced only when galactose is used as the carbon source. So those genes defects you will never get to know unless you use this specific medium where galactose is the only carbon source. Now imagine that you will have a mutation because of which the cell cannot grow in the presence of galactose. As I said that for galactose metabolism, you require a set of enzymes that are not produced in the presence of glucose. Therefore, there is two implicit conclusions from here. One is that something, because of the presence of galactose, sends an information inside the cell that, you know, we are only in presence of galactose, we have to turn on expression of certain enzymes. So that is a signal transduction process or information transmission process, which will definitely involve certain transcriptional regulators. If those are defective, then also you will not be able to produce those genes that are only required when galactose is present. And of course, any gene that is directly responsible for galactose metabolism, they will also, if they are temperature sensitive mutant, then also they will not be able to grow in presence of galactose. So you can, using a temperature sensitive mutation, you can look at all generic things. You'll get a large superset that, you know, these are 500 mutants that cannot grow at an elevated temperature. Some will not be able to grow because of reason A, some will not be able to grow because of reason B, and so on and so forth. Or you can have a very directed search. Okay? If you are looking for, you know, defect in, like if you are trying to identify genes that are important for secretion, you will create an assay only for secretion. So, you know, like this is where the smartness in a genetic screen comes. That what will be your assay so that you can very quickly figure out that this gene or genes, they are defective in my process of interest. Okay, like say if you are trying to look for, you know, a defect in secretion. Okay, secretion means a protein will normally come out of the cell. So if you know a particular protein that comes out of the cell, then you know that you know, like any condition that will prevent that particular proteins coming out of the cell will be involving genes required for secretion of that gene. Okay, you know, I'm saying a little loosely here. It may also be like, you know, a very good example is an enzyme called invertase. Invertase is the enzyme that is secreted out, yeast secretes out invertase only when it is grown in the presence of sucrose as the sole carbon source. So again, there will be some kind of an information transmission that, we are being forced to grow only in the presence of sucrose. Let's produce invertase and send it out. So genes that are involved in production of invertase, if those are defective, then also invertase will not come out of the cell. And if the invertase molecule is produced all right, the protein is produced all right, but somehow it cannot come out of the cell, then there will be a specific defect in the secretory pathway of invertase. And your selection is fairly straightforward, that can the cell grow in the presence of sucrose as the sole carbon source. So in this way, you can actually, come up with screens for, specific aspects of molecular or cellular biology. Okay, and I will test your understanding by setting up assignment questions where you will have to design a screen. Okay, screen design, I am confident that none of you will perhaps ever use an yeast genetic screen in your life. But the thought process that goes into it to design a good screen is pure scientific logic, application of pure scientific logic. That is invaluable. You will need that no matter which kind of laboratory you are in. And that is the reason why I love to test students on their ability to design screens. Okay, so you will have some of those questions that will be there in your assignment. Now I will tell you about one such screen that you can say revolutionary, because it phenomenally increased our understanding of cell biology. And this discovery also earned a Nobel Prize. So Leehartwell, or Leehartwell as he is popularly known, was interested in knowing what are the genes that might possibly be involved in cell division. Okay? So we all know that every different cell type, they have a particular rhythm of cell division. We also know that cell division has different phases, and the morphological description of cell division is very well known. So Leehartwell wanted to know that what are the genes that are involved in regulating this precision in cell division. We are talking about 1967. Okay, this question was asked. In those days, perhaps the scientists did not think beyond their immediate fundamental scientific question. But the discoveries made in those days paved the way for all sorts of cancer drug discovery today. Because in cancer, cell cycle, cell division goes wrong. So this is how fundamentally important this screen is, and you see how simple the logic is. I am biased to think that the more, the simpler the logic of the experiment, simpler the methodology, the take-home message that you get from those kind of experimental designs is phenomenal. They have changed the paradigm, the way we understand biology. Look at it, what Leehartwell did. So he simply grew yeasts and did a temperature sensitive screen, and he identified few colonies that did not grow at higher temperature. Okay? But they did grow a little bit. Okay, like you can take those, but remember you did a replica plating. So in the replica, which is at normal, like a permissive temperature, 23 degrees, they will still have the mutants, the growing. It's very important that you have to have that. Otherwise, you will never be able to characterize it, because dead cells are no good for characterization purpose. So you will go back to the replica plate, and just to remind you that on this plate you see that the colony did not grow, and because it is a replica, so by matching you can say, okay, this is the colony that grew in permissive temperature but not in non-permissive temperature. So you know that in this colony cells have a mutation that does not allow you to grow in non-permissive temperature. So you take those cells, allow them to grow under permissive condition, and then suddenly change the temperature to non-permissive condition. And then look under the microscope, how do they look like? You see here in the top panel, when cells are dividing, you get to see all kinds of budding that is happening. There are different stages of bud formation, cytokinesis and segregation and all of this. Here, you see that the cells do not even look like they are budding. Here, they are clearly budding, but all of them are arrested in a stage like this. You will not find a single one which has a different morphology. So, just by simple examination under microscope, just by looking at it, you can tell that this one has a defect in the budding process per se. And this one has a defect in completion of the budding. Okay? Everything else is done, just the last stage of budding, or last but one stage of budding is arrested. So this is how you can identify genes that are involved in specific aspects of a process. Here it is cell division. You can do it for any metabolic pathway. You can do it for secretion. You just have to think widely and wildly, imaginatively, to come up with a nice way of. You first need to know what is the question that you are asking. Then you need to know, okay, if this is my question, how can I make the cell reveal the secrets? And that's where the entire trick is. The rest is routine. You add a mutagen, grow the cells, they will undergo mutation, and then you flip them and you grow some in, like you make replica, you grow some in normal temperature, you grow some in elevated temperature. These are all routine. Okay? The only trick, the scientific smarts comes or is reflected in how you are going to look at it. And the more you read, you will get to know a whole lot more. Okay? Anyway, so now these kind of experiments, as I referred earlier in lecture 2, is generally done on haploid organisms. Because why don't you do it in diploid? Because see, like this mutant is called CDC28. If it was a diploid, there will be a wild type copy of the same gene, CDC28, written in capital letters, right? And because of that, you will not see the phenotype. Remember, if it is a recessive mutation, then the wild type phenotype shows through because of the wild type copy of the gene. So you will miss all of these mutations. So therefore, these things are done in haploid organism, in the haploid variant of yeast. But with yeast, you have this advantage. You can make them dance to your tune, you can make them haploid, you can make them diploid. What will you do if the organism is diploid by definition? Like say, fruit fly, or mice, or humans, okay? There are much more complex problems if you go to, frogs and zebrafish, 0% zebrafish, they are tetraploid, so forget about it. So in those kind of scenario, scientists have figured out extraordinarily smart ways. Okay, I will not get into the detail, I will just give you the hint of it. That what is the problem? The problem is that any recessive mutation in a heterozygote, you cannot score the phenotype because the wild type copy will mask the mutant phenotype. Okay. So what Nuslin Vollard, she is a legend in, when it comes to discovery of genes and genetic screens, and she figured out her along with Eric Wishaw in the US. They figured out, how the flies are made, literally speaking. And then Nuslin Vollard did it again with zebrafish. Okay, so the philosophy is still the same. That Nuslin Vollard said, okay, I will mutate every fly gene, one gene at a time, and I will see how the fly embryonic development is defective. If I get to see the defect, I'll get to know, what function that gene possibly might be in. The problem is that she is working with a obligate diploid organism. So you cannot do the kind of screens that you did in yeast, because you did it all in haploid. So what she did, there is a concept in fly called balancer chromosome. To be honest, I also do not understand it very well. There are lots of inversions, etc. in a particular chromosome. It is such that it cannot recombine with its homologous chromosome. That's point number one. And then, if the animal is homozygous for the balancer chromosome, balancer over balancer, then it is dead. Okay, and there is one advantage in fly, that there is no meiotic recombination in males, Drosophila males. Okay, so when the meiotic division is happening, if you remember, there can be crossover of genetic material between the mother's copy and the father's copy. But in Drosophila males, that does not happen. So it will not complicate your analysis much further. And fly stocks can be frozen and used much later. Those advantages are also there. So what you do is you have the female, that is, so you have to first kind of know which chromosome, like you have to first determine, okay, that I will do this screen for chromosome number 3. Okay, so you will use a female which is heterozygous for the balancer for chromosome 3. Okay, so here it is the balancer chromosome for chromosome 3, and this is the wild type chromosome for chromosome 3. Okay, then you take the male and you mutagenize it. Not much. You just take the male flies, feed them on the same mutagens, DMSO, like nitrosourea or things like that, and there will be mutations. And then you cross them. Okay. So when you cross them, then in the F1 generation, because you were doing for that particular chromosome, right? So if there is a mutation in that particular chromosome, it will be the mutated chromosome 3 in male over balancer, which came from the female. Okay. So now, because there is no meiotic recombination is possible, so the mutation will stay localized to the original chromosome in the male. Okay, like whatever came from here. Then you cross it. Okay, you take the male and female of this genotype, and then you cross it. Once you cross it, then you get your third filial generation, where there will be some that will be balancer over balancer. Those are dead. You are not worried. There will be some that will be balancer over the chromosome. This is very important. Just like the replica plate under permissive condition was very important, this is very important. You will realize why. And then you will have some which is mutant over mutant. This is where you will see the phenotype, because it is, if it is a recessive mutation, it is homozygous recessive. So you will see the phenotype here. Now, once you see the phenotype here, what if the fly is dead? How will you do the characterization? Then you will go to this fly, which is mutant over balancer, and you do your analysis. Okay, it's a little complicated. Unless you are a dedicated fly person, it is a little difficult. Like honestly speaking, I do not know fly genetics good enough. Okay? But there are some fantastic articles that I will share with you. You should try to read it. Because if you understand fly genetics well, like in my book, if you can understand bacteriophage genetics, there's a beautiful book on lambda genetics. If you understand bacteriophage genetics, if you understand yeast genetics, and if you understand fly genetics, these three, if you understand really well, I mean really, really well, okay, you will be fantastically well prepared to work in any lab where genetics is the mainstay of investigation. And these are not very complicated to learn well. Like for genetics, there are beautiful books. For yeast genetics also, there are very nice review articles that I will share with you. For fly genetics also, there are review articles. The chapter is the art and design of fly genetics, art and design of yeast genetics. I'll share those with you. If you can learn these three, you will be the master of molecular genetic techniques. Okay, there are lots of learnings in there. Anyway, so I don't want to deviate too much. So this is how fly genetics is done. Mouse we will come later. It's a very different ball game. It's done very differently. We'll come to that later. Now let us think back of our temperature sensitive screen. Okay, so you will now have a variety, like if you are trying to, so this is another concept that you should understand. Let us say that there are 10,000 genes in yeast. Okay, in the days when they did the screen, they did not know. Okay, they actually had a much overestimation. So if the way you titrate the chemical for a mutagen, I am emphasizing again that there will be only one mutation per cell and only one mutation, not more than that. That by design it spares most of the cells from undergoing mutations. Okay, so in a plate maybe you get one mutation, two mutation out of 500, 600 colonies. So how many such mutants you must see before you know that you have at least uncovered 20–30% of the genes that are actually involved in a process. That is really complex. So scientists should go for 1 lakh colony, 2 lakh colony, like that, whatever the lab could afford. Okay? And very hard work by lab members. Now, if you have done, say, 1 lakh colony, let's keep the maths simple. So if you have screened 1 lakh colonies, then if in every 500 there is one mutation, so in 10,000 there will be 10 mutations, and in 1 lakh there will be 100 mutants. 100 mutants will fail to grow at 36 degrees centigrade, and they will grow at 23 degrees centigrade. Now, there are two possibilities, that these 100 mutations, all the mutants are in the same gene. That's one extreme. And the other extreme is that in these 100 different mutants, in every mutant, a different gene is mutated. So you have a collection of 100 different genes that are mutated. These are the two extremes. And the real possibility will lie everything in between. That maybe out of 100, 20 will be for a particular gene. The 15 will be for another gene. And for few genes, there will be only one mutant. How would you do it? We are talking about an era where cloning a piece of DNA itself was a 2-year, 3-year project. It was not as simple as today. So how would the scientists do it? They were just brilliant. Okay, they were just super smart. So they argued that if I have 100 haploids which are all mutated in one gene, the same gene, then if I take haploid number 1 and haploid number 2, they will name them somehow. Like here the CDC stands for cell division cycle, because Leehartwell was looking at genes involved in cell division cycle. So he would name the mutants at CDC1, CDC2, CDC3, CDC7, like that, CDC28, CDC3, like that. So let's keep our life simple, CDCX and CDCY. At this point, Leehartwell, me, you, we do not know whether CDCX and CDCY are mutated in the same gene or not. All we know is that neither of these can grow at 36 degrees, but they can grow at 23 degrees. So now I will collect these two clones from the replica in the permissive plate, right? I'll take them, take the haploids. I have to ensure that I have every mutant, both in mating type alpha variant as well as mating type A variant, because two haploids will not mate unless they have opposing mating type. So I will take one in A variant and one in alpha variant. The two haploids, I will mate them and they will form the diploid. I will grow the diploid in 23 degrees centigrade first, and then I will shift them to 36 degrees. If, remember, neither of the two haploids independently can grow at 36 degrees. So if the diploid obtained from there does not grow at 36 degrees, then what does it mean? Okay, let's first look at it here. That the diploid grew well at 23 degrees and it grew well at 36 as well. But the independent individual haploids, they did not grow at 36. What does it mean? Please imagine a little bit, okay? Just a little bit. That in the diploid, one haploid set came from CDCX, another haploid set came from CDCY. So when they are together, imagine the mutation is in different genes. CDCX is mutated in X gene, CDCY is mutated in Y gene. So in the diploid, there is one defective X gene coming from CDCX, but CDCY will supply the good X gene, good allele of X. Similarly, in the diploid, CDCY will supply the defective allele of Y gene, but CDCX will supply the good allele of Y gene. So these diploids are therefore heterozygous for both X and Y. And because these are recessive mutations, the cell does not feel any difference. They grow like wild type. So the defect of X is complemented, the defect of CDCX is complemented by CDCY, and the defect of CDCY is complemented by CDCX. This is called complementation, therefore. This is a complementation analysis. But in the second example, where the diploid failed to grow in 36 degrees centigrade, just like the haploids, chances are that the two haploids are mutated in the same gene. So in the diploid, neither CDCX nor CDCZ could supply a good copy of the X gene. Therefore, it is homozygous mutated for X. I'm not saying it is mutated in the same base. I'm not saying CDCX and CDCZ have identical alleles of the X gene. All I'm saying, CDCX is mutated for X gene, loss of function, recessive mutation. CDCZ is also mutated for X gene, loss of function, recessive mutation. When they are in a diploid, neither of the haploids could supply a good copy of X gene, therefore the diploid also failed to grow at 36 degrees centigrade. So they could not complement. When two mutants cannot complement, they be very careful here, it's tricky. When two mutants or three mutants or four mutants or a group of mutants cannot complement each other for a particular phenotype, they are said to belong to the same complementation group. In those days when the concept of gene sequence, etc., were not really there, scientists got kind of an idea whether it is how many genes they are actually surveying. Like when you have 100 mutations and you do a quick complementation analysis and you formed 10 complementation group, they're reasonably certain that at least 10 genes are involved in the process. There are some ifs and buts in there as well, but largely speaking, you can be reasonably certain that 10 genes are minimally involved in the process of cell division. Okay. So this is the process called complementation analysis. So complementation analysis followed by a traditional forward genetic screen gives you a fairly good idea of how many genes are involved in a particular pathway. And generally speaking, it is always an underestimation. Okay, history has taught us that it's always an underestimation. You get minimally these many genes are involved. Maximally, it's difficult. Like we have learned so much in later years. Okay. I think I will stop here, and then we will start with the new concept in the next class. Okay, that will be lecture number 5. Okay, so with that, I will call it a day today, and I will get back to you again soon.